Hello, everyone, and welcome to today's presentation titled Advance Your Cell Therapy with Innovative CRISPR and mRNA LNP Tools. This webinar is a part of the second annual event in the Cell and Gene Therapy virtual event series. My name is Sydney Panilla, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Okay, now let's get started. I now present today's speakers, Daksha Patel, PhD, Senior Segment Marketing Manager, Cell Therapy, GenScript USA, and Fengmei P, PhD, Head of Product Development and Manufacturing, GenScript Innovation Center. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Okay, Daksha, you may now begin your presentation.
Thanks, Sydney, for introduction, and hi everyone for joining us today. I'm very excited to share how our cutting-edge technology like advanced CRISPR tools and next-generation RNA formats, as well as the LNP delivery, can help to advance your cell and gene therapy project. In this webinar, I will start with the brief introduction about the GenScript, and next I will share the overview on the cell and gene therapy and the CRISPR technology, and then I will cover how our integrated CRISPR toolbox, including the products and service, support both ex and in vivo application. And finally, we will go through a few case study and wrap up with the question-answer session. So let's start with the introduction of the GenScript. GenScript is a New Jersey-based life science CRO with a global presence and offering the wide range of services, product, and instrument. And through our life science group, we help cell and gene therapy developer move smoothly from research to the clinical application. We support academic institution, biotech startup, and pharmaceutical industry in the discovery to development project. And GenScript has built truly a global infrastructure to support the life science innovation. From our headquarters in Piscataway, New Jersey, we have expanded to the key biotech hub across the North America, including the Europe, Asia Pacific, and Australia. We operate the research and manufacturing site located into the New Jersey, Seattle, Nanjing, Singapore, and maintain the regional office on logistic center in the UK, Netherlands, Japan, Korea, China, and Australia. So this is the way we, by our global presence, we ensure that we can deliver high quality product and service efficiently to our customer wherever they are staying and whenever they need any kind of projects and services. So far, GenScript supported our more than 240K customer in more than 100 countries, and our team include more than 500 employees and nearly 10% focus on R&D. And we fill over 480 patent application, and out of them, 260 already get granted. And our impact is reflected in more than 100K present peer review journal. So which citing our GenScript product and services. And this number shows that we are not just the supplier, we are the trusted partner helping the scientists and turn the bold idea into the world impact. So after the brief introduction to the GenScript, let's talk about the cell and gene therapy field. This field is advancing very quickly, and its development happening in many areas, and that's changing how we're going to treat the disease in the wide range of application. So let's take the brief look what's happening in the cell gene therapy area. So far, 36 gene therapy have been approved globally, and including the CAR T cell therapy and 36 RNA therapies. And additionally, like 71 non-genetically modified cell therapy has been approved. So when we're looking at the pipeline, over 4,000 therapies are in development, and nearly half of them is in the gene therapy, and the gene therapy also including the cell modified gene therapy. So sorry, genetically modified cell therapy. And among them, the CAR T cell therapy remain the dominant. And while modalities like TCR-T cell therapy, CAR-NK cell therapy, or CAR-M cell therapy is also growing, and ex vivo delivery is still the most common approach, but in vivo methods are also advancing. So overall, these landscapes highlight how the cell and gene therapy are reshaping the personalized medicine around the world. And many of this therapy optimizing the cell function through genetic modification. And so it's important to improve the efficiency as well as the efficacy, specificity, and therapeutic potency, and reduce the toxicity during this process. So to improve the efficiency and precision, and as well as the safety, safety of the gene editing, ongoing efforts are in progress. And here, this slide illustrates the remarkable evolution happening in the gene editing technology over the past decade. And the key milestone is like scientists have come a long way from early tools like zinc finger nuclease to TALEN to the CRISPR, which made the gene editing faster and more accessible. Here are shown how the CRISPR tools evolve, starting from Cas9 in 2012 for gene knockout, followed by the base editor in 2016 for single base change, and as well as the prime editor in 2019 for more complex edit, as well as the insertion and deletion. And this innovation led to the 2020 Nobel Prize in the chemistry, recognizing the impact of genome editing. And gene editing gene editing tools like keep improving for greater accuracy and fewer off-target effect. And this is the combination of advanced technology and development is already translating into real-world impact. In 2023, Casgevy became the first CRISPR-Cas9-based therapy approved by the Life Science Research Group. And then in December 2024, NK510 received the IND approval in both China as well as US. And this is the allogenic off-shelf NK cell therapy used CRISPR-based cytosine editing. And this innovation shows how the next generation editing tools are moving from the lab to the clinic and unlocking the safe and more effective therapies. So to improve efficacy, specificity, and therapeutic potential, as well as to reduce the cytotoxicity in cell and gene therapy area, different CRISPR tools are required. And there are four essential components required for CRISPR technology. One of them is the editor protein, which is like Cas enzyme or base or prime editor that perform the edits. Another one is the guide RNA, which is direct the editor to the precise DNA or RNA location. And then donor template, which allow the precise edit, especially during the HDR repair pathway. And then to deliver all this system, efficient delivery, different approach scientists using, and that's including like RNP formulation or plasmid, as well as like LNP, mRNA format, cell penetrating peptide, viral vector, exosome, etc. So together, these components form flexible, precise toolkit for both research as well as therapeutic application. And no matter which CRISPR-based therapy researcher developing, success is depend on the right combination of enzyme, guide RNA, and donor template, as well as delivery system, and the quality of that reagents. So GenScript provide all these component in format which is suited for every stage of research, from early discovery to the clinical use, and helping the scientists to move forward with the confidence. At the core of every CRISPR system in the gene editing enzyme is the very essential material. So here is the main CRISPR enzymes listed. One of them is the Cas9, which is classic one, and that makes double standard DNA cut, and it's very good for knockout or HDR edits. Another one is the Cas12, which is similar to Cas9, but it makes staggered cuts and which can improve the precision. The another one, which is more familiar, is a Cas13, which work on RNA instead of DNA and great for temporary editing or RNA knockdown. Then it's coming the base editor, which changed the single DNA later like C to T or A to G without the cutting DNA. And last but not least is the prime editing editor, which is versatile, can insert, delete, or change the DNA without double standard break. So each editor has its own strength, and the right choice depend on your target and the type of edits and the level of precision required for that specific cell and gene therapy. So it's well established that high quality enzymes are key to the success, and through licensing agreement with the Dr. Fengmei lab at the Broad Institute, GenScript bring these innovative CRISPR tools for broader community, and these allow CRISPR tools to be more accessible to researchers around the world. And we provide like high quality Cas enzyme product, including like Cas9, Cas12, Cas13, base editor and prime editor. It's in both the format, like it's protein format as well as in the mRNA format. And we offer the flexibility in the amount as well as the grade. So from RUO to research to full the GMP for clinical use. So this makes easy to scale your project from lab to the work. Here is presented like CRISPR Cas9 performance in human primary T cell across the different quality. So in this experiment, human primary T cell transfected with the different grade of Cas9, and after the transfection, the knockout efficiency was measured using the FACS. And the data indicate that all the three grade, whether it's RUO or basic GMP or full GMP, shows the consistency and high editing efficiency. So all our RUO grade reagents are made using the GMP-like process. So you can start research affordability, means research affordable reagents, and then move smoothly to the clinical grade material. And as your program move from discovery to the preclinical and clinical stage, the need for high quality raw material is crucial. And to meet this demand, GenScript offered the GMP grade bacterial protein expression service, which providing the high quality nuclease such as the Cas9, Cas12, Cas13 base and prime editor, and for various applications. And all our reagents are manufactured under strict GMP guideline and ensuring the aseptic production and full traceability and the regulatory ready documentation. And to date, we deliver over more than 25 GMP batch of the Cas protein, and out of them, like 5 projects already received IND approval. So this track record reflects our commitment to help you move faster and safely from research to the clinic. Next, let's talk about our guide RNA and the molecule which direct the nucleus during the CRISPR use. So GenScript OmniGuide RNA platform support the wide range of CRISPR guide RNA, including the Cas9 guide RNA, prime editing guide RNA, as well as the base editing guide RNA and Cas12 circ RNA. Even we also providing like custom guide RNA for other editing system. So the key feature that differentiate OmniGuide RNA are very impressive because it's offered the longest guide RNA in the industry, up to 266 nucleotide. And all guide RNA are chemically synthesized, which improve the efficiency, reduce the toxicity, and ensure the consistent result. And it supports the phase-appropriate synthesis from RUO to GMP and available in flexible quantities, so starting from 1 to over 1,000 gram, means 1,000 milligram. Even it can go from the gram scale as you go from R&D to the development stage. And last but not least, it comes with the free online design tool to streamline your workflow. So in short, like OmniGuide empower the researcher with reliable, high-performance guide RNA for discovery through clinical development. And in this experiment, like guide RNA are designed for 3 randomly sequenced selected gene target and then synthesized by 3 different vendors, including one of them is a GenScript. And the knockout efficiency was evaluated into the cut cell to compare the performance across the supplier. And the result shows that even the desalted purified form easy edit sgRNA achieve over 80% knockout efficiency for all the target. And this has demonstrated the reliability and strong performance of the CRISPR mediated gene knockout. And GenScript easy edit sgRNA demonstrate the comparable efficiency to the other vendor also. Another one is the safe edit. So here I highlighted the quality of sgRNA can significantly impact your editing efficiency. Safe edit sgRNA have chemical modifications, so that's make them more stable and resistant to the degradation. On the left, you can see the HPLC chromatogram, which shows how effectively truncated sgRNA are removed, which is crucial for minimizing the off-target effect. And we check each batch for purity and consistency using the mass spectrum MS as well as HPLC, and always achieving more than 90%, greater than 90% purity. And on the right side, the bar graph shows the editing efficiency up to 97% across the multiple cell type, including like HEK, A549, and primary T cells. And in short, safe edit sgRNA exhibits high editing efficiency and is a powerful tool for precise genome edit. Now let's talk about the prime editing. It's one of the most exciting frontier in the gene editing. And on the left, we have data showing the dose-dependent editing efficiency using the PEG RNA. We tested the different dose of 141 nucleotide long PEG RNA for both desalt and HPLC purified. And after 3 days using the Sanger sequencing, measure the editing efficiency. And we can see like we achieve with for HPLC purified PEG RNA 63% editing, and even for the 52 with the desalt version. And on the right side, we explore how the PEG RNA length affect the editing. For that, we compare like 141 nucleotide, 150, and 218 nucleotide long PEG RNA. But the 218 nucleotide long version, which include the modification, so it's Q motif at the 3 end and show the highest efficiency. And this modification help to protect the RNA from the nucleus degradation. And this is also observed, supported by the Nelson et al. In their presentation. So GenScript offered the comprehensive tool for prime editing, which is including the off-shelf product as well as custom protein for P2, P3, or Pmax mRNA and LNP formulation. So it's like one-stop solution for prime editing. And here presented like base editing performance in the T cell. On the left side, we used like cytosine base editing and targeting the B2M gene into the cell and achieve high like C to T conversion. And on the right side also we use the another base editing protein, which is adenine base editing, and also target the B2M gene, and the A to G conversion is similarly efficient. So in both the case, we achieve over 90% editing efficiency. And this result demonstrates the precision and high performance of GenScript based editing in the T cell. So in addition to the guide RNA and editor proteins, donor DNA template are also one of the essential for precise genome editing via HDR pathway. So GenScript offered the wide range of high quality HDR template, both in the linear as well as circular format. One of them is the gene exit ssDNA, which is a single standard DNA template and ultra low in toxicity. So it's make ideal for the sensitive application like T cell therapy or generating the cell and animal model. And it can support up to insertion length up to 5 kb. Another one is like gene band dsDNA, is a close and linear double standard DNA that's offer improved stability and integration accuracy and perfect for large knock-in and optimization project up to 10 kb. And then another one is the GenCircle dsDNA, is a small circular DNA vector with no antibiotic resistance or bacterial sequence. So it's non-viral alternative for knock-in antigen expression and scalable solution for high efficiency gene delivery. So all these templates are sequence verified, scalable, and available under the GMP production. So this we ensure that it will bring the smooth transition from research to the clinical application. Here is one representative case study how the GenCircle can work as a apply as a transpons delivery. So the delivery system has a three key component. One is a GenCircle dsDNA, which is act as the vector, and it carries the gene and promoter we want to insert. So suppose here it is used like 3.5 kb CD19 CAR GFP construct, and without because in GenCircle there is no bacterial sequence or antibiotic markers, making it more safe and very useful for clinical application. And the PGK promoter include in the GenCircle to make sure the gene is expressed into the T cell, and then MF transpose enzyme, which insert to the GenCircle into the T cell genome. So together, these three components make up the transpons delivery system, a complete function set up for the testing the gene into the T cell. And using this approach, we achieve about 60% knock-in efficiency, showing that GenCircle is a reliable non-viral method for gene delivery. And now I already mentioned cover so many product features, but let's look at how these tools are making the real world impact. So base therapeutic, which is NK510 program, become the first FDA approval for the base edited NK cell therapy for the solid tumor. And GenScript support the this project of the base therapeutic supported by the GenScript by providing the GMP grade sgRNA, as well as help them to do IND submission. So this demonstrate like our end-to-end support from research to clinical grade material as well as documentation help the pharmaceutical or therapeutic company to go into clinic and help to the patient in real world. So we have many success stories like this, but how GenScript supports in the development of cell and gene therapy, but due to the time limits, I'm not able to share each of them. But if you are interested to know more, you definitely can reach out us. We will give you more information about that. And as August 2025, we deliver over more than 230 clinical grade batch of the sgRNA and supported more than 60 plus IND filing. And out of them, like 18 projects already received globally IND approval, and including 6 get the FDA approval. So this slide also illustrates the full journey from discovery to commercialization. So to support each stage, we offer tailored synthesis solution and the synthesis format. So for REO grade, for early research and screening, and then IND edit grade for IND application and study, and then full cGMP grade for clinical and commercial application. So each format is supported by appropriate quality system as well as compliance for clinical grade material. And as researchers look for more scalable and efficient delivery method, like lipid nanoparticles are becoming increasingly popular, especially for improving the cell viability and it's simplifying the manufacturing workflow. To support this shift, GenScript offers the diverse portfolio of LNP formulation, and it's already optimized for delivering CRISPR component for a wide range of application. So whether you are working with mRNA, sgRNA, or other CRISPR payload, our LNP are designed to deliver high efficiency and reproducibility, and so making them ideal for both research as well as therapeutic development. This slide provides the visual summary of LNP option, the GenScript offering. So GenScript has like all the type of different LNP formulation for Cas9, for Cas12, as well as base editing and prime editing. So altogether, GenScript ready edit offer a flexible, validated, and scalable solution for CRISPR delivery. Here is the one of the case study which highlight the use of dual LNP delivery system to enhance the CRISPR knock-in efficiency. So in this experiment, we use 2 different kind of SM102 base LNP. LNP1, which is deliver Cas9 mRNA and with the sgRNA, and then LNP2 deliver the EGF encoding HDR donor DNA template, which can be either single stranded donor or double stranded donor. And co-delivery of this formulation into the cell, and after that knock-in efficiency measured by flow cytometry after 72 hours. And the data clearly showed that the circular single stranded donor DNA template achieving around 57% knock-in efficiency compared to the linear double-stranded DNA template, achieving around like 35% knock-in efficiency. And circular template high efficiency is likely due to their higher stability. So overall, this dual LNP platform offer a non-viral, scalable, highly effective strategy for precise genome editing for both research use as well as therapeutic application. And one of the biggest challenge in the CRISPR LNP delivery is to payload optimization and delivery efficiency. So that's why our R&D team developed the custom ready edit service, which offering the full support from enzyme selection and then template design to LNP formulation and validation. The process begins with like start phase where you can use the pilot project. Means in your pilot project you use our pre-validated CRISPR LNP system, and then in the build phase we synthesize all the essential CRISPR component like mRNA, sgRNA, DNA, and more, and customize as per your specific need. And then LNP formulation steps focus on the optimizing the delivery. For that, we can provide like recommendation in the component ratio, which help to depend on which kind of application, and that can significantly improve gene editing efficiency and consistency. And finally, in the validation phase, we offer the robust screening platform to test and confirm the performance of your LNP-based construct and ensuring that project is set up for success from the start. So with ReadyEdit GenScript deliver like molecular, flexible, and scalable solution from discovery through the clinical development. So in conclusion, like GenScript is a global life science partner, which is supporting researcher all the way to clinical development. And we providing like everything researcher need for cell and gene therapy development during for gene editing purpose, including like advanced and integrated CRISPR solution, as well as advanced delivery system. So that's way the researcher can move their project efficiently and effect safely from research to the commercialization stage. Now that we already seen how GenScript CRISPR tools are helping to accelerate the clinical program, let's look at the next frontier into cell therapy, which is in vivo CAR-T engineering. So to guide us through this technical detail and delivery strategy, I'm happy to hand it over to my colleague, Fengmei, which is a head of the product development and manufacturing, our RSB Innovation Center. And she will take us deeper into how our platform and innovation solution are supporting in vivo CAR-T cell therapy also. So Fengmei, now you can start.
Thank you very much, Daksha, for your introduction. Good afternoon, everyone. This is Fengmei from GenScript. I would like to talk about mRNA LNP strategies for in vivo CAR-T engineering. In my talk, I will cover about the in vivo CAR-T intro and some progress from GenScript with CD19 CAR mRNA LNP case study, and then our updated advancements in RNA and LNP technologies for your projects. So in vivo CAR-T therapy is the next generation approach where immune cells are genetically engineered directly inside the patient's body, avoid the need of external cell manipulation and the complex lab process. Ex vivo CAR-T mRNA CAR-T therapy traditionally requires harvesting, modifying, reinfusing of a patient's T cell. In contrast, the in vivo CAR-T delivering gene editing tools via viral or non-viral vector to program your T cells inside the body, offering speed and scalability. It reduce the time to treat and manufacturing cost, and also making it more accessible through rare disease, some care settings. To achieve the in vivo CAR-T, LNP is an important tool here to assist with delivery. Comparing the LNP with the AAV vector as another option for the in vivo CAR therapeutic module delivery, the LNP is considered a better delivery vehicle. For the advantages of its efficiency, it can efficiently transfect the T cells, achieve very high transfection efficiency, and there is targeted delivery solution to be achieved with antibody decoration or the formulation optimization, and there is reduced immunogenicity since it can improve the safety profile with the CAR-T therapy and versatility. The LNP can be engineered by its composition optimization to reduce off-target effects and enhancing the immune cell uptake. So looking deeper into the LNP for in vivo CAR-T delivery, it will mainly include these three components. So the LNP formulation that will be mainly modulate its delivery efficiency and side effects. And secondly, the targeting ligands such as CD3 antibody, CD7, CD5 antibody. These are all validated doing pretty well for the T-cell targeted delivery. And last but not least, the cargo. It can be either in the mRNA or circular RNA form, where there is a lot of progress in this field with different companies needing the R&D and clinical phase studies. Looking at this field, the in vivo CAR T is on the rise. There is a lot of pipelines with the leading pharmaceutical companies in different phases of clinical trials. So next, we want to talk about some case study here using CD19 CAR mRNA loaded into LNP for the in vivo CAR T delivery. In this study, we compared how circular CD19 CAR mRNA or the linear RNA perform in vitro first. We used, on the left side, we used the primary T-cell model and look at the CAR expression over different timeline. Up to 76 hours, we could see circular RNA has a great advantage over linear that has a higher expression and a slower degradation over the 96-hour window. Then we look at an in vitro cell model to look at how the CD19 CAR RNA activates the NFAT pathway in a cell model with NFAT leukocyte cat cells and another NLM6 cells. So in this study, we found that the CD19 CAR scarlet RNA has superior performance compared to linear RNA over the 72 hours time window to activate NFAT pathway. We also have a great case study with a customer to look at how the CAR T mRNA in vivo experiments in a mice model. Here, the experiment was using a CD7 antibody that created LNP to deliver the anti-CD19 CAR circular RNA in mice through IV injection, and we observed the body weight and the tumor growth over 14 days. We did to see significant reduction of the tumor growth over the 14 days treatment with the in vivo CAR circular RNA delivery through LNP. Looking further, we want to see how there are more advanced tools to assist with your in vivo CAR T research. So first, let's take a closer look about the RNA advancement for a higher stability. So for the RNA, as we all know, the linear mRNA contains the 5' cap, UTR, and the poly A option. There are different components that we could engineer, including its purification process and the different cap options, and the codon optimization, and poly A tail engineering, and base modification. So here we want to introduce the advancement of the new cap analog, which will enhance the RNA half-life in vivo. There are two cap analog we want to talk about here as we studied in GenScript. One is a 3' ACM modified cap 1. With this study, it showed this modified cap made RNA has slower clearance than natural cap 1 and showed higher translation efficiency than 3' ACA cap 1 in large animals. In a second case, we look at the application of a 2' O modified cap. So the RNA were made with different cap analogs as indicated on the figure legends, and we compared the 2' O modified cap with the 3' ACA cap 1 and the 3' ACM cap 1. The 2' O cap 1 showed even better performance in vivo in mice here, that after 48 hours, we do see the higher expression and the slow clearance of the RNA in mice. Codon optimization for the mRNA is also an important factor for the RNA performance, and we are looked into the AI-powered X5 algorithm for next-generation codon optimization of the RNA for stronger expression and reduce the immunogenicity. Here we compare the different sequences. In the first study, it's a beta-gal mRNA, and we use the gal green assay to look at the RNA expression with different codon optimization. So there is a T sequence, and GSX5 is the sequence used by X5 codon optimization that showed superior expression. And for gene editing application, we also look at compare the Pmax RNA with the GenSmart or the X5 codon optimization. The X5 showed a more superior editing efficiency in the primary T cell assay in this case. We also found this codon optimization could further help with longer half-life slightly for the mRNA. So here is a RLook mRNA. We look at expression with different timeline. Basically, we can see over 48 hours, the X5 codon optimized RNA has a higher expression comparing to traditional GenSmart's optimization method. And reduce the immunogenicity as well. Here we compared the 3 different RNAs. We could see this green bar stands for the X5 codon optimization algorithm should reduce the immunogenicity. Another engineering option is through the tail engineering. We also did this study with poly A-tailed mRNA. We call it stable RNA. So comparing the stable RNA with RNA with normal tail, it showed a higher stability over 48 hours into different assays. Overall, the GenScript has built the platform to provide high-quality mRNA synthesis with different quality grade as a customized solution. And looking forward to working with you on these mRNA projects. We have all the QC spectrum to cover all the purity, impurity assay, and the safety assay details. And as well, we wanted to show that the GenScript has built a strong IP portfolio with the RNA fields with our own UTR patented and co-transcriptional capping method and our codon optimization and poly A vectors. These are also open for customers' projects use for your drug development projects. Beyond that, we also have the circular RNA platform that, as mentioned, it's also a good option for CAR RNA development. The circular RNA we have evolved through 2 or 3 generations. The first generation is the circular RNA with SCAR through self-circularization. And last year, we have updated this with a scarless circular RNA option derived by pi for efficient self-circularization. And we also have a mature platform to make the circular RNA, especially for non-coding RNA through T4 ligation method. These are all good options to consider for the RNA-based therapeutics development. Next, let's look at the technology advancements in LNP section for T-cell targeted delivery. GenScript has built the complete LNP formulation capability with a different scale from 0.1 milligram to gram level, and there is a grade C clean room in our Seattle site to facilitate with your LNP projects with discovery and process development applications. We have validated the LNP formulations here through IV and IM study in vivo in mice, and we have developed optimized LNP for generic use, for targeted delivery, and for gene editing applications. Here is a summary of the list of the customized LNP options we offer. The generic LNP list, we have the standard LNP with SM12, ALC0315, MC3, LP01, and our licensable LNP named CXM2. Welcome to talk with us if you are interested on this one. And the targeted LNP platform, we have two technology mainly. One is using ligand decorated LNP, another is the sort LNP through adding a fifth component into the LNP formulation to achieve spleen, liver, or lung targeting effects. And there is also optimized LNP and N2P ratios for formulation for ready edits for gene editing applications. So here, let's take a look with the standard customer project like SM12 based LNP formulation. In QC, we look at its size, zeta potential, and the PDI. So this we will critically control those under very narrow spec for high quality. And we also look at how the encapsulation efficiency is always more than 90%, and we also analyze the percentage of empty LNP through nano FCM, and it showed the empty LNP in our formulation is less than 2%. Regarding there are two major platforms. One is ligand decorated, one is the sort LNP. So ligand decorated, here we show example with CD3 and the CD34 LNP, which have, since we have antibody available from GenScript, we are also open to accept customer provided antibody for LNP decoration. And there is the formulation optimized LNP, we call sort LNP, for your options as well. Here is some in vivo study data to show different, to look at different formulations biodistribution performance. We injected FLUC mRNA LNP into mice through IV injection at the dose of 0.3 mg per kilogram. And then we look at the FLUC mRNA expression in the whole body, look at after 24 hours on the left. So the left is showing as a log scale bar, and on the right, we look at the ratio of different organs, how the mRNA is expressing. Interestingly, we can see this liver, lung, or spleen sort LNP has achieved a very high organ selectivity. Yeah, regarding ligand decorated LNP, in detail for the chemical ligation route, there are two options for generic antibody decorated LNP. One common way that we have optimized is using the melamide reaction with a SATA modified antibody, which is widely applicable for all antibody LNP decoration projects. Secondly, if you have a specific peptide that could be modified with DBCO or a clickable linker, that will be help with the conjugation to achieve a site-specific modification on LNP. This we have validated in our experiments using this click reaction. It could enhance the targeting efficiency comparing to the non-specific decoration. Some advancement here for the CAR-T delivery LNP solution. We have a new solution called the GSEM LNP that we have found that it enhanced the delivery of CD19 CAR mRNA, especially circular RNA, to gel-cat cells, achieved more than fourfold higher expression comparing to standard SM102 mRNA from Moderna. And regarding antibody decorated LNP, that also greatly enhanced delivery efficiency for T cells. Here we use the primary T cell as a cell model, and we screened the different antibodies. We found the CD3 and the CD7 antibody decorated LNP achieved about 5 or 6 7-fold enhancement of delivery efficiency comparing to non-modified LNP. In conclusion, we would like to introduce the CAR mRNA LNP enables rapid, scalable, and safer in vivo CAR T generation. And for the RNA part, modified cap, modified tail, codon optimization, and scarless circular RNA are the great advancements in RNA technology to enhance CAR RNA expression. In the LNP field, the LNP advanced with optimized formulation, surface engineering with T-cell receptor ligands can further improve T-cell targeted delivery efficiency. We look forward to exploring partnership with you and GenScript for advancing your in vivo CAR T drug development. Thank you very much. Yeah, please let us know if any question.
All right, wonderful. Thank you very much, Daksha and Fengmei, for your informative presentation. We will now go ahead and move into the Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just submit the question in the Ask a Question box located at the bottom of your screen. We will answer as many of your questions as we have time for. Okay, let's go ahead and get started here. The first question that we have says, how do I design my sequence with X5 for codon optimization?
Thank you. I will take this question. So for that, we haven't launched a public tool yet. You can send us your sequence and we will do the codon optimization with our AI team and we'll send you back the sequence.
Perfect. Okay. The next one that we have here says, have you tested other locus for knock-in?
Right. So for the knock-in study, we showed some data with RAB11. I think we also tested some other locuses like track, but RAB11 has shown much higher locking efficiency in the study.
Okay, wonderful. Next question says, how long does the enhanced CAR expression from new capped RNA typically last compared to conventional mRNA?
Thank you. I will look into this question as well. So in our study, we look at the stability over 48 hours. We could see that enhance the expression. But in general, linear mRNA, you would expect its decrease over different time zone. And we can look at the 3' ACM modified mRNA in the longer time course study. It could last for about 48 hours, I would say. There's a significant reduction. But circular RNA has a longer half-life that, as we observed, we could still see good expression at 96 hours.
All right, fantastic. It looks like we have one more here, so we'll go ahead and wrap up with this final question. This one says, what are the key design requirements and testing steps you recommend when developing T-cell targeted LNPs in-house?
Great, thank you. Yeah, I'll think about this question. So for T-cell targeted LNP, I would say T-cell is very sensitive to immune toxicity. Of course, the safety is the primary consideration. And secondly, delivery efficiency, like an decorated LNP showed a significant enhanced delivery compared to non-decorated one. I would say these are the two primary questions we want to take consideration when design the LNP, and then go to detail. We would like to optimize the entropy ratio to achieve very good encapsulation efficiency and the homogeneity of the LNP to achieve high delivery efficiency.
All right, fantastic. Well, thank you again, Daksha and Fengmei, for your time today and for your very important research. Before we go, I would also like to thank the audience for joining us today, and I would like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall following the webinar for more information and resources. Questions submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. As a reminder, this webinar can be viewed on demand for 1 year until October 15th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time, thanks so much, everybody. Goodbye.